Get ready, everyone, for another deep dive into the origins of life. This time, we're going to be tackling a paper by Søren Toksværd that proposes a pretty fascinating and maybe even unexpected order for the milestones that led to life.
Yeah, a really interesting take.
As we know it, we're talking about the transition from a prebiotic world
Right.
to one teeming with complex, self-replicating organisms.
Exactly. So Toksvær's paper lays out this compelling argument
Okay.
for a specific sequence of events, and it suggests that homocularity in proteins actually came first,
Oh, wow.
followed by the development of metabolism and genetics, and then finally the emergence of cells.
That's different from what we're used to hearing about.
Yes, it is quite different from some other models,
Yeah.
and it definitely has some intriguing implications.
Okay. Before we get too far into this, though, can you remind us why homocularity is so crucial
For sure.
for life? It's a concept I've always found fascinating,
Yeah.
but it's worth revisiting its significance.
Absolutely. Homochirality, it refers to the handedness of molecules. Lots of organic molecules, like amino acids and sugars, they exist in two forms that are mirror images of each other, just like your left and right hands.
Yeah.
They have the same chemical formula, but different spatial arrangements. Now, for life as we know it, this spatial arrangement is critical
Okay.
because enzymes, the proteins that catalyze almost all biological reactions, are highly specific. They'll only interact with molecules of a particular chirality.
So if a molecule has the wrong chirality, the enzyme won't be able to interact with it effectively.
Exactly.
And the biological process it's supposed to facilitate just won't occur. It's like trying to fit a left-handed glove on your right hand.
Precisely. And this is why homochirality, where biological systems consistently use one chiral form over the other,
Yeah.
is so fundamental. It allows for the precise and predictable interactions that are absolutely essential for complex biochemical pathways to function.
Okay. So homocularity is key.
Hmm.
But why does Toksværd argue that it came first, even before things like metabolism and genetics? This is where the paper really piqued my interest.
Well, Toksværd makes a pretty compelling case based on the inherent stability of homochiral peptides,
Okay.
which are chains of amino acids. He points to this concept of chiral discrimination, which essentially means that homochiral peptides are more likely to form stable structures in environments with low water activity. They can arrange themselves into those classic alpha helices and beta sheets, which are way more stable than the structures that would be formed by mixtures of left and right-handed amino acids.
So in the prebiotic world, with its potentially limited water availability in certain environments, these homochiral peptides would have had a survival advantage,
Yes.
persisting longer, having more opportunities to interact and assemble into more complex structure.
Exactly.
It's almost as if the very nature of these molecules predispose them to be the building blocks of life.
That's a great way to put it. Their inherent stability under those early conditions would have given them a head start, setting the stage for the development of more complex systems. And here's where it gets even more interesting. Toksværd connects this back to aging and mortality. He suggests that even in modern organisms, these very stable protein structures are susceptible to a process called racemization over time.
Racemization. Can you elaborate on that for our listeners?
Sure. Racemization is the process where those perfectly ordered homochiral structures start to kind of break down. They revert to a mixture of left and right-handed forms. You can think of it like a carefully organized library gradually falling into disarray. Toksværd specifically mentions aspartic acid, an amino acid that's particularly prone to racemization as a potential marker of aging in everything from bacteria to humans.
So the very stability that allowed these homochiral peptides to thrive in the prebiotic world also makes them vulnerable to this gradual breakdown over time. It's a fascinating link between something as fundamental as the handedness of molecules and the universal experience of aging.
It really is, and it raises these profound questions about whether aging is truly an inherent part of life itself, like baked in from the very beginning.
Okay, so we've got this world of homochiral proteins, but life needs more than just stable structures. It needs energy, and it needs instructions to truly take off.
Right.
This leads us to the next major milestones in Toksvær's model,
Yeah.
the emergence of metabolism and genetics. And here's where things get a bit more complicated, right?
Yeah, the relationship between metabolism and genetics is a bit of a chicken or the egg scenario, which came first. Did metabolism need pre-existing genetic information to guide its pathways, or did genetics emerge from a pre-existing metabolic network?
A classic dilemma. I'm curious to hear how Toxviate approaches it. Let's
hmm
start with metabolism.
Okay, so the paper discusses the foremost reaction. This is a process that can create carbohydrates spontaneously from simpler molecules like formaldehyde, which were likely abundant on early Earth. However, there's a catch. The Formose reaction, at least as we've observed it in the lab, produces a mixture of both D and L forms of carbohydrates.
Oh, so we run into that chirality issue again. And if I remember correctly, life as we know it exclusively uses D carbohydrates.
You are absolutely right. So the question is, how did that transition from a mixed pool of carbohydrates to the exclusive use of D forms occur? And this is where enzymes, those highly specific proteins we were talking about earlier, come back into play.
So are we suggesting that specific enzymes capable of distinguishing between D and L carbohydrates must have emerged really early on, even before metabolism became fully established?
That's the implication, and it's a fascinating one. Toxfair argues that the presence of these stereospecific enzymes, even in the most ancient life forms that we know of, suggests that they must have been present in that prebiotic world,
Oh.
influencing the direction of metabolic pathways from the very beginning.
That's a really thought-provoking idea. But how could just a few enzymes have such a profound impact on shaping an entire metabolic system? It seems almost too simple.
It might seem simple, but don't underestimate the power of enzymatic catalysis. Imagine a single enzyme that just slightly favors a reaction involving a D-carbohydrate.
Okay.
Over countless reaction cycles, that small bias could have a cascading effect, leading to a system where D-carbohydrates become the dominant form.
Like a domino effect, with that first enzyme setting off a chain reaction
Exactly.
that ultimately leads to the establishment of homochirality in carbohydrates.
But where does genetics, the other piece of this puzzle, fit in?
So genetics likely co-evolved with metabolism. Remember, DNA and RNA, those are the molecules of inheritance, and they also rely on D-carbohydrates, D-ribose, and D-deoxyribose, to be precise. So the same selective pressures that favored D-carbohydrates in metabolism would have also favored them in those emerging genetic systems. So we've got this incredibly intricate interplay between homochirality, metabolism, and genetics. It's as if these fundamental aspects of life were bootstrapping each other into existence.
Precisely. And this brings us to the final milestone in Toxford's model, the emergence of cells,
Okay.
which represent yet another leap in complexity.
Yes, we've gone from simple molecules to complex networks of reactions or information storage.
Right.
But how do we get from there to enclosed, self-replicating units that we recognize as cells? That seems like a giant leap forward.
It is a significant leap, and one that requires a different set of explanations.
This is where I think we should pause for now, letting our listeners absorb these complex ideas before we delve into the final stage of Toxford's model.
As we were discussing before the break, the emergence of cells from that prebiotic world of molecules and complex reaction networks represents a major transition in the origins of life. It kind of requires us to consider a different set of mechanisms than those that we've been exploring so far.
Yeah, definitely. We've talked about the inherent stability of homochiral peptides, the emergence of metabolism and genetics, but those processes, while complex, still lack that defining characteristic of life as we know it, compartmentalization.
Exactly. The ability to maintain a distinct internal environment separated from the outside world,
Right.
that's crucial for life. This allows for the controlled regulation of biochemical processes, the concentration of reactants, and the maintenance of gradients, all essential for the complexity of life. So how do we get from a world of molecules to a world of enclosed, self-replicating compartments?
The billion-dollar question, isn't it?
Yeah.
I'm very curious to hear how Toxford tackles this challenge in his model. It seems like a big leap from the relatively simple principles of chirality and molecular self-assembly that we've been discussing to the intricate architecture of a cell.
It is a substantial leap, and Toxver draws on recent advances in our understanding of self-organization and non-equilibrium systems to bridge that gap. He proposes that a process called non-equilibrium phase separation played a crucial role in the emergence of the first cell-like structures.
Non-equilibrium phase separation. Okay, that sounds a little technical. Could you break that down for our listeners? It's not a term we use every day.
Of course. It's actually simpler than it sounds. Think about oil and water. They don't mix, right? They spontaneously separate into distinct layers or phases. That's what we call equilibrium phase separation, a system settling into its lowest energy state.
Right, like a vinaigrette salad dressing separating if you leave it on the counter.
Precisely. Now, imagine a system where there's a constant input of energy, preventing it from reaching that equilibrium state. That's what we mean by a non-equilibrium system. And in those systems, you can get much more interesting and complex behaviors emerging.
So it's not just things settling out according to their inherent properties.
Right.
It's a dynamic interplay between those properties and the energy flowing through the system.
Exactly. And this constant flow of energy can actually lead to the spontaneous formation of compartments, even from mixtures that wouldn't normally separate under equilibrium conditions. In the context of the origins of life, Toxvard suggests that these non-equilibrium conditions might have been present in certain environments on early Earth, driving the formation of those first cell-like enclosures.
So instead of a static primordial soup, we're imagining a more dynamic, energized environment where these protocells could emerge. But what kind of environments is he picturing? What could have provided this crucial energy input?
Toxvard points to what he calls hot spring environments, those dynamic interfaces between the Earth's crust and the early oceans. These areas would have been rich in chemical and thermal gradients, with hot fluids from the crust mixing with cooler ocean water. These gradients would provide a constant source of energy, driving the system away from equilibrium and potentially creating the conditions for non-equilibrium phase separation to occur.
So those classic images of primordial soup might not be entirely off the mark,
Yeah.
as long as we envision them as more dynamic, more energized, perhaps with volcanic vents or geothermal activity churning the metaphorical pot.
Yeah.
But even with these compartments forming, how do we get from simple enclosures to something that resembles a true cell? Something capable of carrying out all those complex processes that we associate with life.
That's where things get even more interesting. Toxfair draws on recent research showing that active chemical reactions within these compartments can actually drive their growth, their movement, and even their division.
Right.
It's not just about passive encapsulation, it's about the internal chemistry actually influencing the behavior of the compartment itself.
Wow. So it's almost like the chemistry is becoming self-organizing, almost like a primitive form of metabolism directing the behavior of the protocell.
Yeah.
But what kind of reactions are we talking about here? How could they possibly lead to something as complex as cell division?
So this is where the details get a little more speculative. We're kind of pushing the boundaries of what we currently know. But Toxford points to research showing that certain reactions, especially those involving polymerization and depolymerization, can generate forces and changes in osmotic pressure within droplets.
Polymerization and depolymerization. Could you remind us what those terms mean? Some folks might not remember their high school chemistry.
Oh, absolutely. Polymerization is the process of linking small molecules, called monomers, together into long chains called polymers. And depolymerization is just the reverse of that process, breaking polymers back down into monomers. These processes are fundamental to biology, and they involve changes in energy and molecular organization that can influence the physical properties of the compartment they're happening in.
So you're suggesting that these pretty simple processes of building up and breaking down molecules could have been enough to generate the forces needed for those early protocells to actually grow and divide.
That's exactly what he's suggesting. And while it might seem a little counterintuitive, just think about the incredible diversity and power of chemical reactions. If these reactions were coupled to the energy gradients present in those hot spring environments, they could have provided the push and pull, the dynamism needed to drive those primitive forms of growth and division.
It's a remarkable concept. So instead of relying on intricate molecular machinery like modern cells use for division, these early protocells might have been utilizing much simpler chemical processes, driven by the energy flows in their environment. It's almost like they were dancing to the rhythm of the Earth's internal heat. But are we talking about true cell division here, with the replication of genetic material, all that complex coordination, or something more primitive?
It was likely a simpler form of division, maybe something more like budding or fission, where the protocell basically splits into two without the precise control of genetic replication that we see in modern cells. But it's a crucial step towards the complexity of life as we know it.
Right. A fascinating bridge between the molecular world and the cellular world. But before we move on, I want to circle back to something you said earlier about aspartic acid and racemigration. You suggested that it could be a marker of aging, even in modern organisms. I'm curious to explore that idea a bit further. How does this process of racemization, this gradual breakdown of homochirality, actually manifest itself in living things?
That's a great question. While the precise mechanisms and implications of racemization are still being investigated, there's growing evidence suggesting that it contributes to the aging process in a bunch of different ways. For example, racemization can actually alter the structure and function of proteins, leading to a decline in their efficiency or even making them totally inactive.
So those very carefully crafted homochiral proteins that are so crucial for life's processes gradually lose their structural integrity and their ability to function properly.
Right.
It's almost like they're slowly unraveling over time. But are there specific examples of how this might actually affect the aging process?
Certainly. One example is the accumulation of D-aspartic acid in the brain. Aspartic acid is an amino acid that plays a role in various neuronal processes. However, as it racemizes to its D form, it can build up in certain brain regions, and that can potentially interfere with normal neuronal function. This accumulation has been linked to age-related cognitive decline and even neurodegenerative diseases like Alzheimer's.
So it's not just a theoretical concept.
Yeah.
It's an actual process that might be contributing to some of the most profound challenges we face as we age. Are there other examples, perhaps in other parts of the body?
Yes, there's evidence that racemization affects a variety of tissues and systems. For example, in the lens of the eye, the accumulation of racemized aspartic acid can cause changes in protein structure and aggregation, which can contribute to the development of cataracts. And similarly, in the skin, racemization can affect the elasticity and structural integrity of collagen, which is a key protein that gives skin its youthful appearance.
So we're seeing this process at play throughout the body, impacting everything from our cognitive abilities to our physical appearance. It's a bit of a sobering reminder that even the most fundamental aspects of our molecular makeup are subject to that relentless march of time. But wouldn't natural selection favor organisms that could, resist or reverse race discrimination? Why haven't we evolved better mechanisms to prevent it?
Well, that's a question evolutionary biologists have been wrestling with for decades.
Right.
And while there's no one definitive answer, there are several interesting hypotheses out there. One possibility is that the energy cost of completely preventing racemization might just be too high. Organisms have a finite amount of energy,
Right.
and they have to allocate it carefully to various functions like growth, reproduction, and repair.
So it's a trade-off. Investing energy in preventing racemization might take away from other essential functions, but wouldn't the benefits of a longer lifespan outweigh those costs?
Well, that's not necessarily the case. Remember, evolution operates on the time scale of generations. If an organism can survive long enough to reproduce and pass on its genes, any additional investment in longevity might not give it a significant evolutionary advantage.
It's a humbling reminder that evolution doesn't necessarily strive for perfection, but just for good enough, as they say.
Mm.
But there's another layer to this, right? Toxford himself suggests that racemization, and by extension aging, might not simply be a consequence of wear and tear,
Right.
but actually an inherent part of life itself. It's a thought-provoking idea that kind of challenges our typical view of aging as something to be fought against.
It really is a thought-provoking perspective, and it kind of aligns with that whole concept of entropy, that tendency for systems to move towards disorder over time.
Yeah.
If homochirality represents a highly ordered state, then racemization, that gradual shift towards a mixture of chiral forms, could just be seen as an inevitable consequence of the laws of thermodynamics.
It's almost like life is in this constant battle against entropy, trying to maintain this really delicate balance of order and disorder. And while it might seem counterintuitive, maybe there's something beautiful in that. It reminds us of our connection to the rest of the universe, subject to the same fundamental forces.
It does. It suggests that aging might not be an enemy to be conquered, but just an integral part of that grand cycle of existence.
That's a profound thought, one that I think deserves more reflection. But before we dive too deep into those philosophical implications, I think we should take a step back and kind of synthesize what we've learned so far. We've covered a lot, from the fundamental importance of homochirality to the emergence of metabolism and genetics, and finally that remarkable process of protocell formation.
Yeah, we have. We've seen how each of those milestones builds on the one before it, creating this complex and interconnected web that ultimately led to the emergence of life as we know it.
And we've explored this fascinating connection between homochirality, racemization, and aging, raising questions about the nature of life itself, the inevitability of aging, and our place in the universe.
A truly insightful journey so far.
And there's still so much more to explore. When we come back, I want to dig a little deeper into Toxford's specific ideas about where these crucial steps in the origins of life might have happened. Where on early Earth could these remarkable events have unfolded? Welcome back to our deep dive. We've been following Søren Toxford's model for the origins of life, a pretty unique sequence of events, and now I want to focus on Location.
Okay.
Where on early Earth could all this have happened?
It's a key question, and Toxavir tackles it head-on in his paper. He actually proposes distinct locations for different stages in the origins of life, each one offering specific conditions that would favor those particular
So
processes.
not just one primordial soup where everything happened at once, more like different settings for
Exactly.
different acts in the grand drama of life, so to speak.
A great analogy.
Can you elaborate? Where does he suggest these key events might have taken place?
Okay, so for the initial stages, the emergence of homochirality, metabolism, and genetics, Toxavir suggests the Earth's crust.
The crust. Not the first place I'd think of. We usually picture those warm little pools on the surface. Why the crust? What advantages would it offer?
Well, the crust, while it seems harsh, actually offers some advantages for those early stages. First, it's more stable than the surface, which would have been bombarded with radiation, meteorites, wild temperature swings. The crust provides some shielding from those harsh conditions.
Makes sense considering how fragile those early molecules and proto-life processes would have been. But were the chemical ingredients even available in the crust?
Oh, absolutely. The crust is rich in those essential elements needed for life. Carbon, hydrogen, nitrogen, oxygen, phosphorus, all the building blocks. They would have been present in the minerals and fluids within the crust, providing the raw materials for life's chemistry to get going.
Interesting. It challenges that classic primordial soup idea. What about water, though? We think of life being so dependent on water.
Right.
Would there have been enough in the crust?
That's key. While the crust wouldn't have had those vast oceans like we see today, there would have been water trapped within its structure, within the mineral matrix. This water, often in thin films or small pockets, would have provided that aqueous environment needed for the reactions to happen.
So not a giant ocean, but more like a network of microenvironments within the crust,
That's right.
each with those crucial ingredients and conditions.
Exactly. And these microenvironments would have offered a whole range of conditions, different temperatures, pressure, pH, chemical composition. That diversity might have actually been beneficial, providing a sort of natural laboratory where all sorts of combinations of molecules and reactions could be tested out.
Like a natural selection process, but on a geological scale, with different environments favoring different chemical pathways.
I like that, a natural laboratory.
But we've talked about how water activity is so important for homochiral peptides, both for their stability and their vulnerability.
Yeah.
Wouldn't the lower water activity in the crust make those peptides super stable? Maybe even too stable for life to evolve.
That's a great point. Low water activity does favor the stability of those initial homochiral structures. But as those structures get incorporated into protocells and eventually true cells, they need a more dynamic, watery environment for all those complex reactions to happen.
So a shift from that static crust environment to something more dynamic is needed,
Right.
something more suited to those intricate processes of life. Is that where those hot spring environments come in?
Exactly. For cells to emerge, those self-contained units capable of growing, dividing, replicating Toxverd suggests those hot spring environments we discussed earlier would have been perfect.
Those dynamic interfaces between the crust and the early ocean, with all those gradients. It makes sense that they'd provide the energy and the right conditions for those protocells to form and take those early steps towards life.
Absolutely. Those hot springs would have had this constant flow of energy and materials, driving the system away from equilibrium and creating the conditions for non-equilibrium phase separation, that potential mechanism for protocell formation we talk about.
Such a dynamic picture. Challenges that old view of life arising in a calm, shallow pool, more like a dance on the edge of a volcano.
Yeah, a very dynamic picture, and it's supported by a lot of evidence. From those extremophiles that thrive in harsh environments to the hydrothermal vents teeming with life on the ocean floor, we're seeing that life is much tougher and more adaptable than we once thought.
And it shows how important it is to consider a wide range of environments when thinking about the origins of life, not just those that seem comfy to us. Opens up possibilities for where else in the universe life might exist.
Exactly. It's a good reminder of how vast and diverse the universe is, and the incredible potential for life to exist in forms and places we can't even imagine yet.
Leaves us with a sense of awe and wonder. Well, I think we've covered an incredible amount in this deep dive. From basic molecules to the emergence of complex systems, to that remarkable transition from a world of molecules to a world of cells.
Yeah, it's been quite a journey.
And we've explored those big questions about the nature of life, aging, our place in the universe, even if we don't have all the answers.
Right.
We've gained a deeper appreciation for the story of life and how we came to be.
Absolutely. And maybe even more importantly, we've been reminded of how much we still don't know and the amazing potential for new discoveries as we keep exploring the mysteries of the universe.
Thanks for joining us on this deep dive. And to our listeners, we hope you've enjoyed the journey and are inspired to keep exploring the natural world. Until next time, keep questioning, keep seeking, and keep being amazed by the incredible story of life.